Very good morning, students. Last class we are discussing regarding the flagella. So cilia and flagella. In cilia and flagella, the how does the movement takes place and what are the structure when you take the cross section of the cilia and flagella, how the organization structure which is arranged we are discussing. Generally, first flagella, as I already told, it is a protozoan or locomotory organism. So they are long, stiff filamentous structures which are always formed from the basal bodies. So this arrangement we are telling 9+2 arrangement. In the structure, if you see, it shows 9+2 organization. So 9+2 organization means how does the microtubules are arranged in the peripheral regions, and in the center 2 microtubules are arranged. That I will draw you and show you how does the arrangement takes place. So we can see in the electron microscope the arrangement is 9+2 array. So that 9+2 array arrangement, how it is, let us see. So, if you take this, totally there are 9 pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 pairs. So, that is the reason the microtubule, the peripheral microtubules, which are 9. So, we call it, and center 2 are there. So, that is the reason we call it as a 9+2 array. (9+2) array. So here the center microtubules are 2, whereas 9 pairs in the peripheral. So this is what we call it as a 9+2 array in the arrangement of the cilia and flagella. So this is plasma membrane. Plasma membrane. Next, these are peripheral microtubules. Peripheral microtubules. That is, those are pairs, no? So they are doublet. We call it as doublet. As they are in pairs, we call it as a doublet. And center, these are peripheral microtubules. Here, how many are there? They are not in pairs. They are separately present. So these two are separately they are present. So they are central microtubules which are singlet. Central microtubules, singlet. So these are attached, the peripheral microtubules. So if you take each one, they have a names of A and B. A, B, A, B. So microtubules are A, B. So for every A attached to B, there will be A, B. Okay. Right. So here, these in turn they are attached. B microtubule attached to A. Okay. So these are what are the attachments which are present? Those are called interdoublet bridges. These are called as interdoublet bridges. Interdoublet bridge, because these are called doublets. So between them, between the doublets, the attachment is called as interdoublet attachment. And we see the spoke-like structures. Thus, to the central microtubule, if you see, from the A microtubule, there will be projections to the center. So such kind of attachments. So these are called as radial spokes. They are called as radial spokes. Radial spokes. Okay. So this center part, we call it as central sheath. This is called central sheath. Central sheath. The central sheath which separates the singlet and the doublet microtubules. Okay, right. So this is the structure of the cilia and flagella cross section. If you take axoneme or axial filament, structure is this. Okay, axoneme, we call it as axial filament or axoneme. Axial filament or axoneme. Okay, right. Next, if you see, there are nexin or the connections for the A microtubule, which is having the interdoublet bridges. So here, if you take, there are certain outer spaces. This is a cell membrane, and you have here the doublet bridges, and there is a gap, again central vacuole, again to this center they are thing. So now the two central microtubules are attached by bridge. This is called as bridge. Okay, note down. Bridge. Next, dynamics are present too. There are small projections called as dynamics on the A microtubule. For every A microtubule, there are apart from the radial spokes, there are dynamics. I'll tell you all the functions of this, how they are present, and what is the reason for this. Okay, so dynamics. Let us see the point wise what is the use of all these things. Okay, so as per the structure, if you see, this is about the cilia and flagella. That is what we call it as axoneme or axial filament. One filament, if you take one flagella, if you take and cut it, you see inside the cross section. This is the thing, sir. Okay, right. So now note down the the 9 plus 2 arrangement. Now you understood what is 9 plus 2 arrangement. Total 9 doublets and 2 singlets. So we call it as 9 plus 2, which are located from the periphery and the central. Okay, so we call it as 9 plus 2 array. So note down, continue the points. The two central singlets continue after the 9 plus 2 array. The two central singlets, each with one microtubule. The two central singlet, each with single microtubule. The two central singlets, each with a single microtubule, enclosed by fibrous inner sheath. Enclosed by fibrous inner sheath. So we saw the structure now. So that is the central sheath which covers those central singlets. 9 peripheral doublets form a cylinder. 9 peripheral singlets forms a cylinder. 9 peripheral singlets forms a cylinder. You saw the peripheral structures now. They form a cylinder between inner sheath and outer sheath. Between inner sheath and outer sheath. Between the inner sheath and outer sheath. Right. Next. Central tubules are connected by bridges and enclosed by central sheath. The central sheath, two central microtubules, that is the central tubules are connected by bridge. The two central vacuoles, the two central tubules are connected by bridges and enclosed by central sheath, that already we discussed, and enclosed by central sheath. Okay, right. Next, central tubules are connected. The central tubules are connected to one of the tubules of each periphery by the radial spokes. Till now we saw how does the central to the central sheath and the peripheral, the radial spokes are connected. So these radial spokes, 9 radial spokes are present. So the central tubules are connected to, note down, the central tubules are connected to one of the central tubules connected to one of the tubules of each peripheral. Connected to one of the tubules of each peripheral. One of the tubules of each peripheral. Okay, that is peripheral, you have what? Doublets. Each peripheral doublets. So that is with the radial spokes. So what was we by a radial spokes? Okay, right. Next. So these are totally how many? 9 radial spokes by a radial spokes. How many total? 9. So 9 radial spokes. So that is what the radial spokes. The peripheral doublets are also interconnected by linkers. The peripheral doublets are also interconnected by bridge. The peripheral doublets are also not bridges, linkers. In turn, they are connected by the peripheral microtubules are in turn connected by linkers. So what are these peripheral doublet names are given? They are AB linkers. These are called as AB linkers because A is connected to B, as I already show you. A is connected to B, so we call it as AB linkers. Okay, right. Or interdoublet bridge. Interdoublet bridge. You can call it as linkers or interdoublet bridge. Okay, right. So this is with regarding to the cilia and flagella. So write down some points with regarding to cilia where they are present. So they are nothing but the cilia act like a oars. Cilia act like a oar. In paramecium and all, if you see, the total body is covered with the cilia. Structure is one and the same for the cilia and flagella, but the long bristles, these are short. So what does the cilia? They are oar-like structures. No doubt. So this is explanation of the structure, that is axoneme. Cilia, these are oars. These are just like oars. So that is nothing but the paddles. The oars are nothing but the boat. How you move the boat when you ride with a paddle? So that is just like those oars for movement of cell or surrounding fluid. For the movement of oars, for the movement of cell or surrounding fluid. Okay, right. Next, these are short hair-like present all over the surface of the body. Cilia are short. They are, you see projections in the case of hair-like structures. They are short, hair-like. So present all over the surface of body. Present all over the surface of the body. Present all over the surface of body, or you see specific species, specific regions of ciliate protozoans, or specific regions of ciliate protozoans. Alright, right. Next, The paramecium, if you take, they have cilia all over the body surface. Paramecium have cilia all over the body. Cilia have all over the body surface, you will be having it. Right. So this is about the cilia and flagella. So we have completed the next cell organelles, that is cilia and flagella. Now similarly we see such kind of a structure where you have centrosome and centrioles. Centrosome and centrioles. So that is also how does the cross section we see the structure, that also we have to discuss. So those also have microtubules and all. So next cell or keep the heading centrosome and centrioles. Centrosome and centrioles. So centrosome is discovered by Boveri. Scientist Boveri. The centrosomes are discovered by scientist Boveri. These are, what are centrioles and centrosome means? The centrosome is an organelle which has two cylindrical structures which are arranged at right angles to each other called as centrioles. So that means centrosome consists of two centrioles. Each centrosome consists of two centrioles. So they are present. These are two cylindrical structures arranged at right angles to each other. That is, two cylindrical structures arranged right angles to each other. Right angles to each other. Okay, right. Next, they are surrounded by pericentriolar materials. These centrioles are surrounded by pericentriolar materials. Pericentriolar material. So what is this in structure? We will see what are all these pericentral material and how does these centrioles are arranged. So they are non-membranous. Seen in pairs and self-duplicating units. Centrioles are non-membranous and self-duplicating. They are non-membranous and self-duplicating structures. They are present in animal cells except in RBC, erythrocytes. They are present in animal cells except in RBC. erythrocytes, except in the RBC erythrocytes. Okay, next. These are made of protein tubulin and small amount of ATPase. Made of protein tubulin and ATPase enzyme. So tubulin proteins, those are nothing but the microtubules. Tubulin proteins or microtubules. Right. So if you see the structure, then you can clearly understand how does the arrangement it is. Okay. So let us see the structure of this centrosome and centrioles, how they are arranged. Right. So here also, when you take this is also the two sets. See, generally the centrosome, if you take it is see, suppose this is a centrosome organelle. You see the arrangement of two cylindrical structures are perpendicular to each other like this. So this is what the centrioles. So the two centrioles. So this is centrosome. Centrosome having two centrioles, that is right angles to each other. Okay. So this, for each one, how it is observed, how it is seen, we are discussing in this particular picture. So these are cylindrical structures which are having microtubules like this. But when you see from this section cutting, when you cut it and see how the inside arrangement is there, how does those 9 are arranged, we are seeing. So here also you have a peripheral microtubules, but they are not just like in the case of this, they are arranged as the 3 microtubules. So peripheral microtubules, the peripheral cylindrical microtubules. So these are 3 triplet, 9 triplets are present at the periphery. So 9 triplets are seen towards this, 2 pairs, sorry, 2 triplets, 3, 4, 5, 6, 7, 8, 9. So like this you have 9 triplets. So just if you think previous one, that is cilia and flagella, here also they are 9 triplet microtubules, peripheral. 9 triplet microtubules which are peripheral. Previously in cilia and flagella only doublet, but here triplet. So for that also, here also you have names of ABC. Okay, ABC for that. Though there you have AB, here you have ABC. So all are having a common wall. So they are fused with a common wall. They are fused with a common wall. But center, you don't have any microtubule. This is a space called as a central hub. Central hub. Okay. And just like in that particular cilia and flagella, here also they are interconnected to the spokes. These doublets, they are interconnected to the spokes to the center. So like this, to the center microtubule, they are arranged. So if you see this structure, then the link here, these are also interlinked with the linkers. So the structure is same, those linkers. So here if you see, these are called AC linkers. There it is called as AB linkers, here it is called as AC linkers, or sometimes they are also called CC linkers. They can link to C and C or A to C linkers. So these are microtubules, 9 peripheral microtubules, which are triplet. Okay, right. Next, these are tubulin proteins. Microtubules are tubulin proteins. They are made of tubulin proteins. Right. So this is the arrangement. So as there are no microtubules at the center, we call this as a 9+0 array. Very important. Why it is called 9+0 array? Because the center microtubule is absent, so we call it as 0. Peripheral 9 triplets are there, so we call it as 9. So it is just like a cartwheel shape. Okay, it is just like a cartwheel shape. So that we see the space inside, we call it as a matrix. So we see the matrix which is present inside the hollow space. Okay, right. So here this is pericentriolar material. Because on the surface, if you see, they have these are with here they are totally surrounded by macules. Macules. All these functions I'll tell you. We call it a pericentriolar satellite. Pericentriolar satellite. Macules are pericentriolar satellites. Here you can clearly okay. Macules are pericentriolar things. So these are two centrioles. So these are the centrioles. So let us see in points what are the functions of all these things which are arranged. Okay, right. So note down, these are also radial spokes. Radial spoke. So the structure, just we'll explain about the structure point wise. So it is a cartwheel structure of the structure so here if you see, it is a cartwheel appearance. it appears as a cartwheel. Okay, that is spokes, hub and peripheral fibrils with spokes, hub, central hub, no? Central hub. The spokes, central hub and peripheral fibrils and peripheral fibrils together it appears like a cartwheel. So the microtubules, the 9 triplet microtubules are there, no? Peripheral microtubules, they are hollow tube open at both ends. The 9 peripheral microtubules, are hollow and open at both the ends. Open at both the ends. Okay. Next, these are made of tubulin protein. They are made of tubulin protein. The angle between them is 40 degrees. Generally, if you see, they are parallelly, 9 triplets are running parallel to each other, parallel longitudinal to each other with an angle of 40 degrees. Arranged parallelly and longitudinally at an angle of 40 degrees. Okay, so that is what the arrangement with an angle. So the condition is called, so 9 plus 0 condition. We call this as 9 plus 0 condition because of central part does not have any fibrils. 9 triplets and central microtubule is absent. Microtubule. Okay, right. Does not have any fibrils. Right. So it does not have any microtubule. So that is why we call it as 9 plus 0 condition. Okay, right. Next, each one linkers, how does they are linked? So the CA linkers or AC linkers, that point you note down. The microtubules are arranged with the peripheral microtubules, because there are only peripheral microtubules in the flagella, in the centrioles. So the microtubules, peripheral microtubules are linked. Commonly we have connections. The triplet fibrils, that is our microtubules. What do you call it as other name, like the triplet fibrils or microtubules. These are connected by AC linkers or CC linkers. Just now I show you in the diagram, one peripheral triplet to the other peripheral triplet it is attached with the AC linkers. And the spokes, radial spokes are CA linkers, strands that connect the CA linkers with radial spokes. The strands connect which connect so to the that connect CA linkers with radial spokes. CA linkers with radial spokes. Okay, right. Now we discussed about the massieules or pericentriolar satellites, what we are telling. So they are 9 in number. The massieules are what do you call it as? Pericentriolar satellites. Are 9 in number. They are 9 in number. And what is their function? The function as nucleating centers for the formation of astral rays. For the formation of astral rays. And function as nucleating centers. And function as the nucleating centers for the formation of astral rays. For the formation of astral rays. See, astral rays, you know, in the process of cell division, from the microtubules which are at the periphery, astral rays are formed. So if you see the diagrammatically, during the cell division in the animal cells, the astral rays are generated. So these astral rays are first formed from the peripheral material, that is the massieules, satellites, and those in turn will form the filaments. So astral rays are formed a star-like appearance during mitosis. So that only forms the spindle fibers. Later spindle fibers will originate from this for the attachment of microtubules. So this in detail we will study in the cell division chapter. Okay, so they form the astral rays and more centrioles. So centrioles are formed from these massieules. So what is the function of these centrosome and centrioles? They form basal bodies which gives rise to cilia and flagella. They form basal bodies which gives rise to cilia and flagella, which gives rise to cilia and flagella. Okay, and these only will form the spindle apparatus during cell division. They form spindle apparatus during the cell division. So they gives rise to spindle apparatus during cell division. So in mitosis you see only one spindle apparatus. So how one cell divides into two cells. With the formation of one spindle apparatus, we see one cell divides into two. Okay, a cell is dividing into two means with one spindle apparatus, that is through formation of spindle fibers, prophase, metaphase, anaphase, telophase and all these things, one spindle apparatus. Whereas in the case of meiosis, if you take two, how? First it forms one cell, diploid cell forms two haploid cells, and each one again will divide into 2 cells. Okay, so here totally, if you see, there are 3 spindle apparatus. Here 1, which is dividing 2, again here 2, here 1, 3. So like that, when you call it a spindle apparatus, in meiosis, if you take 3 spindle apparatus are formed, in mitosis only 1 spindle apparatus is formed. So this is about the centrosome and centrioles. Okay, right. So next class we are left with microbodies and nucleus. Okay, so next 1 or 2 classes we will complete this particular chapter also. Okay, right. Okay, so see you in the next class. Thank you.